the keynote gets you the videos because, I want you to have clean copies to use. And I apologize, there are 6 of those in there, but I find that that does a really good job of giving people other connections to things. And I'm sharing things with you that I've not shared before. And I think that this is outstanding timing wise because tomorrow in Oslo, I believe they're having a review, one of the courts in preparation for ICC for holding these people criminally accountable.
Well, that is absolutely, well, it's not wonderful. I don't know if I can say wonderful to that. But,
no, you have to, because at this point in time, since war has been launched, responding back to it to hold them accountable is what you need to do. It's kind of like Nuremberg 1. This is going to be Nuremberg 2.0. And I'm helping them behind the scenes, and I will be one of the expert witnesses if it goes to the ICC.
Well, anything that I can do to assist in that process in whatever way I can, I want to do that. So let's get rock and rolling. We are in record, and I'm recording a couple of different places, and we'll just get on with it. Dr. Fleming, welcome to BASES 2021. It's an honor to meet you. Go ahead, sir.
It's my pleasure to be here, Miles. Thank you so much. I'm going to share the screen and begin the presentation. Well, Miles and everyone, hello. This is Dr. Fleming from the Dallas-Fort Worth area. It's my pleasure to present how SARS CoV2 gain-of-function research violated international law for the 2021 BASES Summer Conference this 11 July 2021. As always, I want to mention briefly the conflict of interest that I am the patent holder for something called Fleming method or FMTVDM, and the author of the inflammation and heart disease theory that explains how inflammatory diseases like heart disease and a variety of other diseases are associated with a number of factors, including bacteria and viruses, including SARS CoV2. We're going to cover 6 topics today. We're going to look at how the United States paid for this gain-of-function bioweapon. We're going to look at this bioweapon for what it really is, a prion bioweapon, with the virus SARS-CoV-2 merely being the delivery system. We're going to look at how do we know what virus we're actually dealing with. We're going to ask the questions, how effective are the vaccines really, and are they needed? We're going to look at treatments for this bioweapon. And finally, we're going to address the very important issue of Nuremberg 2 for holding the people accountable, responsible for the funding, development, and dissemination of this bioweapon. Research in the United States can be dated back to 1999 when the Health and Human Services provided funding for infectious diseases of coronaviruses. A year later, in 2000, Ralph Baric began the initial process of using reverse genetics to rescue an infectious clone known as SARS-CoV-2, something that will be very important as we get through the presentation. Within 2 years, colleagues of Ralph Baric, including Curtis and Young at the University of North Carolina, obtained a patent that was paid for by NIH grant GM 63228 for recombinant engineering or reverse genetics for the development of infectious, replication defective coronaviruses. Also in 2002, Dr. Shizeng Li at the Wuhan Institute of Virology received funding and proceeded to develop infective coronaviruses, including the use of HIV pseudovirus glycoprotein 120, a prion in its own right. The following year, in 2003, Ralph Baric received another NIH grant, allowing him to synthetically alter coronaviruses or coronaviridae. The first notation in the published literature of SARS-CoV-2 was not in 2019, but actually back to a Chinese paper in 2006. And any place that you see asterisks, as you see here, you'll see an associated publication at the very bottom. Throughout these slides, you will be able to look up for yourself these actual published papers and read more detail on your own. In 2007, the National Science Foundation published research that would have been well known to anybody in infectious disease in 2019. That research project looked at the best plan of action for a pandemic. This was done at Indiana University and institutions in both Italy and France. And the conclusion was you must immediately shut down international travel if you believe you have a pandemic. 4 years later, in 2011, the world became painfully familiar with gain of function research as labs at the University of Wisconsin and in the Netherlands allowed to get out a very infectious gain of function research virus for the H5N1 Asian avionic influenza flu. The following video is a discussion that Dr. Anthony Fauci is having with participants looking at gain of function research, and this conference was done in 2012.
Integral to that study has always been the issue of gain of function research, not only for influenza, but essentially for all infectious diseases research. Now, there are a few ways to look at gain of function research. There's the naturally occurring mutations which naturally give gain of function, and investigators study these effects on the phenotypes of interest. Does this mutation make something more transmissible, more pathogenic, or adapt to host better? Or, what historically investigators have done is to actually create gain of function by making mutations, passage adaptation, or other newer genetic techniques such as reverse genetics and genetic reassortment. When we do that, often some phenotypes appear and others disappear. For example, it is commonly seen that when you increase a transmissibility, you may see a decrease in pathogenesis, or vice versa. You may deliberately increase pathogenesis and see a decrease in transmissibility. But the bottom line is that gain and loss of function research is critical to understanding disease pathogenesis, antimicrobial resistance, and host responses, as well as to developing better techniques of surveillance, vaccines, and therapeutics, specifically to gain of function research on HPAI H5N1.
Okay, because you've got the video, and you know who that is. That is Dr. Anthony Fauci in 2012 dealing with the consequences of this. In 2013, only a year later, another coronavirus was well documented for fatalities in Saudi Arabia and South Korea, and that virus is known as the Middle Eastern Respiratory Virus, or MERS. We learned from research during that period of time that treatment of these viruses were quite doable and manageable. However, it required immediate treatment upon symptoms of individuals. Barrick and several Chinese physicians at the time began to isolate several coronaviruses of particular interest, those with what's known as an HKU4 spike, which up until that point in time had been unable to actually infect people, although their future work would change that. By 2014, the CDC had another problem with gain of function and release of infectious pathogens, where several of the CDC workers were accidentally exposed to anthrax. Again, there was shipment of deadly influenza virus, and the NIH discovered 50-year-old vials of smallpox not kept in isolated areas that they had forgotten about. At that point in time, in 2014, the Obama administration called for a halt to gain of function research. However, this particular video shows one of the reporters asking Dr. Anthony Fauci about gain of function research and the need for control of where US government funds are being delivered so that those monies don't fall into the hands of people who might be doing something more nefarious.
So I have a question about the role of dual-use research developing super influenza viruses in developing vaccines. We interviewed Ron Fouchier, who sort of is the guy who exploded some or brought this to a lot of people's attention. And I accept that he's confident and his lab probably will protect us. The only problem, and any BSL-4 has the ability, the only problem is that humans work in labs. And we know from anthrax that humans sometimes err even if all the systems are perfect. So could you just give a sense of the role of this kind of research in developing vaccines and whether we've got the kind of, whether we actually can ever have controls which would protect us?
No, when you say I want to make sure that I'm answering the question that I think I'm hearing.
Yeah.
You know, when you mention Ron Fouchier, you're talking about gain of function. Like what role of gain of function? You know, there is, I mean, if you completely ban all gain of function research, then you're going to interfere with some of the important things we need to do. But there's some gain of function research that you really don't need to do. So I think that we have to, and we've struggled with it for years. It isn't perfect yet. To have some guidelines, and again, you could only control what the federal government funds. You can say we're not going to fund any research that puts the possibility of danger of an escaped microbe only if you fund it, because you can't control what private industry is going to do unless you make a law to prevent them from doing that, which I think is not going to go a long way. Bottom line, I don't have a good answer for you, except that when we talk about those kind of experiments, as we've struggled with over the years, we try to create a culture of responsibility. And when, you know, culture of responsibility is as good as the people who are responsible. And we saw, as you said, with anthrax, that there are crazy people around who do crazy things. And I don't think we're ever going to get away from that, no matter what you do. So there's no good answer to it except to say to try and create a culture of responsibility when we do these things. Sorry, I couldn't give you a more precise answer. That's usually not the way I answer a question. Is kind of just the duration of the answer is disproportionate to what you know about it.
So I guess the answer is we should still worry, but work even faster for universal flu vaccine, which even gets enhanced. Okay, thanks.
Sorry.
Also in 2014, it became painfully obvious that the United States had funded research for gain of function of coronaviruses. This is the actual international patent itself that was issued to Dr. Ralph Barrick at the University of North Carolina in 2014. And as you look here at the lower area, it specifically states for the methods and compositions of chimeric coronavirus spike proteins. Chimeric is the scientific term for combining more than one organism together to make something new, and that is gain of function. You can see here that this was paid for, funded by National Institutes of Health, which are U.S. taxpayer dollars. In 2015, Shi Zhengli, referring back to the HKU4 spike protein from a couple of slides back, had designed and worked on this spike protein for the purpose of making it infective in human beings. And there were two specific mutations that they did at that point in time to make the HKU4 spike more infectious. Barrick of the University of North Carolina and Shi Zhengli of the Wuhan Institute of Virology, all funded by Peter Daszak of EcoHealth that received funding from U.S. federal government sources, announced that they could make a more dangerous, virulent, and infective virus. And in fact, the following year in 2016, they published a paper that you can see here was funded under the acknowledgments by NIAID, the National Science Foundation of China, the USAID EPT Predict, which is our CIA and Department of Defense, EcoHealth Alliance, North Carolina, University of Texas, and a variety of other federal funds. And you can see to the left the actual changes in this virus. The bottom part shows the different components of different viruses that were put together including the ASHC014 spike protein. And you will also see 5 specific inserts of specific nucleotide bases that were changed. So this is not just the spike protein of one virus added to the backbone of another, but it has 5 additional specific insertions, the latter one being in the envelope region, which is critical for infection and transmission of this virus into the brain to cause brain infections. Despite this research, the gain of function research ban was lifted in 2017. And the following year, Shizheng Li gave a well-attended presentation at Shanghai University in November on the studies on bat coronaviruses and its cross-species infections. That data has since been deleted, as was the data bank for the Wuhan Institute of Virology during the summer of 2019. Prior to the outbreak in December, and the report by the Wuhan Municipal Health Commission report regarding that outbreak has also been deleted. This particular video is held between an Italian investigator and journalist discussing Peter Daszak and Ralph Barrick at EcoHealth and the University of North Carolina, and their connections with Shizheng Li at the Wuhan Institute of Virology.
Nel 2015 la professoressa Shi si rivolge ad uno dei maggiori esperti nella costruzione di virus sintetici. È il professor Ralph Barrick dell'Università del North Carolina, il più grande esperto al mondo di coronavirus. Insieme i due scienziati costruiscono un virus ibrido, la famosa chimera di cui aveva parlato il TG Leonardo, tornato a circolare in rete qualche tempo fa.
Un gruppo di ricercatori cinesi innesta una proteina presa dai pipistrelli sul virus della SARS ricavato da topi e ne esce un supervirus che potrebbe colpire l'uomo.
2017 il team di Wuhan pubblica le sequenze di 11 nuovi coronavirus di tipo SARS, identificati ancora nel sud della Cina, nello Yunnan. Ricombinando alcuni di questi virus in un progetto sempre cofinanziato dal governo americano, i ricercatori costruiscono 8 diverse chimere. 2 di loro sono in grado di infettare le cellule umane. La prova, ancora una volta, che i coronavirus dei pipistrelli sono pronti a fare il salto senza bisogno di passare da un altro animale.
There were 2 teams in the world who were very good at making chimeric viruses, and they were both working on SARS-like coronaviruses. One in North Carolina under Ralph Barrick, and one in Wuhan under Shizheng Li, and they both developed techniques for combining 2 different parts of different viruses into one virus, the backbone of one virus and the spike protein from another virus. These experiments were being done to understand how virulent, how dangerous coronaviruses were, particularly SARS-like coronaviruses, in order to be able to be ready to combat a pandemic. They warned the world in a publication in 2015, jointly between Professor Barrick's team, Shizheng Li's team, that they were capable of making more dangerous pathogens and that this was a risky line of research.
In total, EcoHealth received more than $61 million in federal funding from the United States of the monies that I'm aware of. And you'll notice that of that 61 million, more than half of it came from the Department of Defense, with the second largest contributor being Health and Human Services. In addition to the monies that were provided to Peter Daszak at EcoHealth, the Department of Defense also provided a policy advisor by the name of David Franz. Franz had been the former commander of Fort Detrick and involved with research in biological weapons. This video is Senator Dr. Rand Paul asking very specific questions regarding gain of function of Dr. Anthony Stephan Fauci, as Dr. Rand Paul is attempting to find out what Dr. Fauci knew about monies that were being sent from the United States to Peter Daszak at EcoHealth, Ralph Barrick at the University of North Carolina, and Shizheng Li of the Wuhan Institute of Virology.
Dr. Fauci, do you still support funding of the NIH funding of the lab in Wuhan?
Senator Paul, with all due respect, you are entirely and completely incorrect that the NIH has not ever and does not now fund gain-of-function research in the Wuhan Institute
Do they fund
of Virology.
Dr. Barrick?
We do not fund.
Do you fund Dr. Barrick's gain-of-function research?
Dr. Barrett does not doing gain-of-function research, and if it is, it's according to the guidelines, and it is being conducted in North Carolina.
You don't think inserting
China.
a bat virus spike protein that he got from the Wuhan Institute into the SARS virus is gain of function?
That is
you would
not
be in the minority because at least 200 scientists have signed a statement from the Cambridge Working Group saying that it is gain of function.
Well, it is not. And if you look at the grant and you look at the progress reports, it is not gain of function, despite the fact that people tweet that.
So do you still support sending money to the Wuhan Virology Institute?
We do not send money now to the Wuhan Virology
Do you support
Institute.
sending money? We did, under your tutelage. We were sending it through EcoHealth. It was a sub-agency and a sub-grant. Do you support that the money from NIH that was going to the Wuhan Institute?
Let me explain to you why that was done. The SARS CoV2 originated in bats in China. It would have been irresponsible of us if we did not investigate the bat viruses and the serology to see who might have been infected.
Or perhaps it would be
China.
irresponsible to send it to the Chinese government that we may not be able to trust with this knowledge and with this incredibly dangerous viruses. Government scientists like yourself who favor gain-of-function research.
I don't favor gain-of-function research in China. You are saying things that are not correct.
Government defenders of gain-of-function, such as yourself, say that COVID-19 mutations were random and not designed by man. But interestingly, the technique that Dr. Barrick developed forces mutations by serial passage through cell culture that the mutations appear to be natural. In fact, Dr. Barrick named the technique the no-see-em technique because the mutations appear naturally. Nicholas Baker of the New York Magazine said nobody would know if the virus had been fabricated in a laboratory or grown in nature. Government authorities in the U.S., including yourself, unequivocally deny that COVID-19 could have escaped a lab. But even Dr. Shi in Wuhan wasn't so sure. According to Nicholas Baker, Dr. Shi wondered, could this new virus have come from her own laboratory? She checked her records frantically and found no matches. That really took a load off my mind, she said. I had not slept for days. The director of the gain-of-function research in Wuhan couldn't sleep because she was terrified that it might be in her lab. Dr. Barrick, an advocate of gain-of-function research, admits the main problem that the Institute of Virology has is the outbreak occurred in close proximity. What are the odds? Barrick responded, could you rule out a laboratory escape? The answer in this case is probably not. Will you, in front of this group, categorically say that the COVID-19 could not have occurred through serial passage in a laboratory?
I do not have any accounting of what the Chinese may have done, and I'm fully in favor of any further investigation of what went on in China. However, I will repeat again, the NIH and NIAID categorically has not funded gain-of-function research to be conducted in the Wuhan Institute of Virology.
This particular video looks at Senator Kennedy as he's asking point-blank questions to Dr. Anthony Fauci and Dr. Collins of NIH, asking how they can be certain that the money that was provided from those agencies didn't fall into the wrong hands, producing a nefarious gain-of-function virus.
Madam Chairman, Chairwoman. Dr. Fauci, I believe you have testified that that you didn't give any money to the Wuhan lab to conduct gain-of-function research. Is that right?
That is correct.
How do you know they didn't lie to you?
Excuse me, sir?
How do you know they didn't lie to you and use the money for gain-of-function research anyway?
Well, we've seen the results of the experiments that were done and that were published, and that the viruses that they studied are on public databases now. So none of that was gain of function. So
how do you know they didn't do the research and not put it on their website?
there's no way of guaranteeing that, but in our experience with grantees, including Chinese grantees, which we've had interactions with for a very long period of time, they're very competent, trustworthy scientists. I'm not talking about anything else in China. I'm talking about the scientists, that you would expect that they would abide by the conditions of the grant, which they've done for the years that we've had interactions.
So you don't think the Chinese would lie to you?
Well, when you say the Chinese are a rather broad group. I know the scientists that we've dealt with have been trustworthy.
Mm-hmm. You think all the scientists have told the truth in terms of the origin of the Wuhan virus and not been influenced by the Communist Party of China, do you?
I don't have enough insight into the Communist Party in China to know the interactions between them and the scientists, sir.
Right. Why are we giving them money in the first place?
Well, that's a very good question, and thank you for giving me the opportunity to answer it. Well, SARS CoV2 started in China, in Guangdong province, and it went from a bat to a civet cat to a human.
Yes, and excuse me, Doc, for interrupting you, but our time is so
limited. No, I'm going to be real quick.
Our time is so limited. Why are we giving money to the labs in China to study virology?
Well, I'm going to give you a rather succinct answer to that, sir.
I appreciate that.
And that's why I was saying the SARS CoV2, clearly the bats that have the viruses, that are the coronaviruses, are in China. As I said a couple of times, it's not in Fairfax County, Virginia, or is it in New York? It's in China. So if you want to show and study, importantly, the animal-human interface.
Because that's where the bats are.
Yeah, the bats. That's where the bats are.
I want to be sure I understand your testimony. You didn't give money to the Wuhan lab to do gain-of-function research.
That is correct.
And you believe they didn't do gain-of-function research because they told you they didn't.
We've seen the results of the studies that they conducted. And they were not
including any private studies.
Excuse me? Including?
Any private studies.
I'm not sure what you're getting at, sir.
Here's what I'm getting at. You gave them money and you said, don't do gain-of-function research.
Correct.
And they said, we won't.
Correct.
And you have no way of knowing whether they did or not, except you trust them. Is that right?
Well, we generally always trust the grantee to do what they say, and you look at the results.
Have you ever had a grantee lie to you?
I cannot guarantee that a grantee has not lied to us, because you never know.
Yeah. Can we agree that if you took President Xi Jinping and turned him upside down and shook him, the World Health Organization would fall out of his pocket?
I don't think I can answer that question, sir. I'm sorry.
Well, do you think that the President Xi Jinping has undue influence over the World Health Organization, do you?
I have no way of knowing the influence of the president of China over the WHO.
Okay, so you think the WHO is a completely independent body, level playing field, call it like you see it, and they really want to get to the bottom of the origin of the virus. Do you believe that?
My interaction with the WHO and for Dr. Tedros, the Director General, has been one that I do believe he's a person of high degree of integrity.
I want to ask one last question. Why did you guys spike, not guys and ladies, why did y'all spike the prior administration's investigation into the origins of the coronavirus and whether it could have come out of the Wuhan lab?
Sir, we did not spike anything in the prior administration. I'm not sure what you mean by spike, but we have no influence.
The State Department spiked the prior administration's study.
But that has nothing to do with the National Institutes of Health.
So they didn't consult with y'all?
They did not.
Did they consult with you, Dr. Collins?
I read about it in the press this morning.
Doc? They just spiked it without talking to their experts? You don't want to answer that one, do you?
I just read about it.
Thank you, Madam Chair.
Senator Murphy.
Thank you.
What we can take away from this gain-of-function virus and the money that was used to develop it is that it had a significant impact on the entire planet. As we can see from June 23, 2021, and these numbers keep growing larger every day, this virus, SARS coronavirus 2, and the impact upon health, governments, lifestyles, the entire planet has been substantial. There is no country, no peoples that were unaffected by this. With, at the point of June 23, more than 180 million cases around the world and almost 4 million deaths. With tens of millions of dollars paid out by the federal government from taxpayer dollars, from the FDA, the National Institutes of Allergies and Infectious Disease, Health and Human Services, the National Institutes of Health, and the U.S. Department of Defense. Monies that over the course of decades went through NIH and NIAID, many times under the direct supervision of Dr. Anthony Fauci. Monies that went to Peter Daszak of EcoHealth, and later Ralph Barrick at the University of North Carolina and Shizhang Li of the Wuhan Institute of Virology. A bioweapon was made. A bioweapon that has impacted the entire planet. What we want to look at now is what type of bioweapon this really is. And while many people are thinking of this as a virus, the virus is merely the delivery system for the real bioweapon, a prion. And a prion is an abnormally folded protein that causes damage throughout the body, including mad cow disease, Alzheimer's, inflammation, and blood clotting. SARS-CoV2, the abbreviation for Severe Acute Respiratory Syndrome Coronavirus 2, is the 7th known coronavirus that's been able to infect human beings. We talked about 2 others earlier, SARS-CoV-1 and MERS. These viruses, as you can see, are extremely small, 80 to 160 nanometers, and a nanometer is 1 billionth of a meter, 10 to the minus 9 meters. This cryo-electron micrograph allows you to actually see what these coronaviruses look like with the crown shape, which are nothing more than the spike proteins sticking out around the edges of the virus itself. Our real focus needs to be on the gain-of-function spike protein. And here you can see 3 specific regions outlined in this spike protein. The bottom part here shows the membrane of the virus, and the tree-like structure is the actual membrane or molecule that makes up the spike protein. And there are 3 specific areas to pay attention to. The first one is the HIV pseudovirus glycoprotein 120 that Shizhang Li previously mentioned she had been working with to insert into coronaviruses. The second one includes 4 amino acids known as proline, arginine, and alanine. Every amino acid is given a different letter of the alphabet for abbreviation purposes. And so proline, arginine, and alanine are referred to as PRRA inserts. And they form what's known as a furin cleavage site, which increases the infectivity. The consequence of these insertions into this spike protein molecule is much like stepping on a box. If once you step on it, it changes the shape of the box. So an action on the molecule changes the shape. The insertion of the HIV glycoprotein 120 and the PRRA insert have produced a change in the spike protein itself. And at the very top, where the spike protein attaches to the ACE2 receptors on our cells, that's known as the receptor binding site because it's the receiving part of the spike protein by our cells. That area has been changed to develop a prion-like domain. And that prion-like domain behaves like a prion, and as you'll see, is associated with neurologic disease and cardiomyopathies or heart damage. Luc Montagnier, the man who received the Nobel Prize for discovering HIV, and Jean-Claude Perez have gone to an extensive amount of work to look at the spike protein itself and to look for evidence of HIV insertions. This schematic with all of the red arrows shows you how much HIV has been inserted into the spike protein. There's more than 18 RNA fragments inside the spike protein, which match either HIV, human immune deficiency virus, or SIV, simian or ape immune deficiency virus. It's interesting to note that Luc Montagnier also served as a research professor, a university chair at the same university in Shanghai that Dr. Shizheng Li had given the presentation to in 2018. And in addition to Dr. Sheng Li's presentation no longer being found on the internet or the university site, the information regarding Professor Luc Montagnier's being a university chair there has also been deleted from the university website. Dr. Luc Montagnier and Jean-Claude Perez also provided additional information about the PRRA insert that we talked about previously. Each amino acid requires 3 nucleotide bases, so the PRRA insert is 12 nucleotide bases being inserted. Mutations occur typically 1 at a time, not 12, but it's important to note that Luc Montagnier and Jean-Claude Perez have found 1,770 nucleotide bases within the spike protein that match HIV. The reason why HIV glycoprotein is so critical is that it's a prion. We know that this particular part of HIV causes neurologic damage. Back in the 1990s, I began looking at what's called NUR5AC, which is N-acetonuramic acid. This is very unique to human beings, and it is the reason why certain types of animal foods like beef and other foods are associated with an inflammatory disease in people, because we don't possess the enzyme capability to break down those meats, and it produces an inflammatory reaction in people. One of the reasons we believe that I was targeted early on was that the research work that I did with this sialic acid raft, critical for this glycoprotein 120 to cause prion diseases, and my exposing the misinformation by Big Pharma on the use of radioactive nucleotides, and the incorrect use of the nuclear cameras, is that I was, at that point in time, unwittingly beginning to expose much of the work that was going on by these researchers back in the early 2000s and late 1990s. Here's an example. The black and white images, A and B, show a number of areas that are white, which appear like a sponge. This is the result of HIV glycoprotein 120 being exposed to brain tissue. It produces these sponge-like changes. That sponge-like change in medical terms is referred to as spongiform encephalopathy, or brain that looks like a sponge. In lay terms, that's referred to as mad cow disease. On the right side of the graphic, you'll see B and E. E is the normal neurons shown with the green fluorescent, and B shows what happens when that same tissue is exposed to HIV glycoprotein 120. the neurons die and cease to be able to produce the components to keep the nerve cells active. In addition to the HIV glycoprotein 120, the PRRA insert can be looked at here in a variety of ways. The insert is actually on the stable part of the spike protein, and the spike protein can be divided into the S1 and the S2 attachment. The furin cleavage site, which is shown here, which is the PRRA furin cleavage site, is in the stable part of the S protein, S1 region. So there's no reason to expect any type of mutation to be showing up there. The arrow off to the right shows a list of coronaviruses, none of which except SARS CoV2 contain the PRRA insert, as shown by the entire list of coronaviruses. This is critical for this virus to infect, as shown in an article in Molecular Biology, where they note that the furin cleavage site, this PRRA, is essential for this virus to be taken into the cells as well as it is. In fact, it increases by a fold of 10 to 20 times the infectivity of this spike protein. And interestingly enough, the government that owns the patent rights for the furin cleavage site enzymes is the United States, as shown here in this patent from 2007. Also noted here, you'll see that this furin cleavage site is critical not only for the infection of the virus, but it's critical for working with HIV and glycoprotein 120. It's also critical for suppressing something known as P53 enzyme system, which prevents humans from developing cancer. So the presence increases the ability of the glycoprotein 120 to exist and exert its effect, and increases the likelihood that people will develop cancers. Finally, remembering back, as I said before, the insertion of the PRRA and the HIV glycoprotein 120 insert resulted in a change to the spike protein molecule. We see the prion-like domain shown as number 3. There's two sets of images here. One shows the results of, let me see if this works. There we go. At 2 weeks, humanized mice ended up dying after they were exposed to this spike protein. And that produced changes in the brains of those mice, producing that same sponge-like brain, producing mad cow disease. In fact, it was found in all of the animals. Rhesus macaques were also studied using this same approach and killed after 5 to 6 weeks. And in their brains, in those areas as shown there, what we saw was an inflammatory change as shown by the T cells and all the activated microglia, as well as the Lewy bodies, which are a type of prion disease. And that disease is associated with Alzheimer's and a number of other neurologic diseases. These are slowly progressing diseases that do not occur overnight. It takes a period of time for them to develop. And previously, they have been untreatable, and one would say lethal, because of the damage and the debilitation that these prion diseases produce. Well, how do we know what virus we're actually dealing with? Well, we come to the term polymerase chain reaction, or PCR testing. That's how we know what we're dealing with. And Harry Mullis developed this patent and received it a couple decades ago. And in the upper right-hand corner, you'll see the number of cycles required to actually run a PCR test. And you'll see a red rectangular box showing that Harry Mullis in the patent said that we stop at 20 cycles, because as you can see there, that reproduces something more than 1 million times. In fact, it produces 1,048,555 copies. The threshold for doing this test and knowing whether you have something valid is therefore set at 20. And if you look over at the left-hand part, you'll see that that threshold cycle of 20 is circled in two places, showing that that is more than sufficient to reach the threshold level to show what you're looking for. And this is done by having a forward primer and a reverse primer, which we're going to talk about in a slide, and a fluorescent component, which is reproduced in the appropriate genetic area. And it's the fluorescent compound that triggers the activation for a positive test. And so PCR allows us to look for a genetic code to see if something is present. And you can tailor make that specific code for what you're looking for, which is how it's been used for SARS CoV2, because we know the genetic code of SARS CoV2. But done correctly stops at 20 cycles, because anything else you can see has an exponential phase that just blows it out of the water and makes it completely useless as a test. So how do we use it? We begin with a forward primer, which tells the test where to start looking for the correct genetic code. And you can see all sorts of sequences here: T-thymine, A-adenine, C-cysteine, G-guanine. These are the DNA bases. U for uracil is for RNA. And the forward primer says, start looking here once you've reached that. And it requires a match of somewhere between 18 to 24 nucleotide bases to be to start reading. And then the reverse primer tells you where to stop reading, and you reproduce in those areas where you find a forward primer and reverse primer in your product, and you do the cycles up to 20. And also in here, then you put the fluorescent stain compound to give the light signal for the PCR test to actually read. There is a difference between those genetic bases and something like the end product, which is the protein in the case of the vaccines, or in the case of the virus, the spike protein. And so we have a couple terms to learn. One is genomics, or the genes. That's the DNA and the RNA. And the process of going from DNA to RNA is called transcription. You're transcribing from one language, DNA, to another language, RNA. And that's different from what's called proteinomics, or protein. And the protein is made by taking the RNA and translating it to a protein. Now, how is this done? Well, quite simply, you take 3 of the nucleotide bases in the mRNA, and 3 of those bases together is called a codon. And each codon, as you can see in the middle part here, with first base, second base in the codon, and third base, when put together in sequence, give you a specific amino acid that then, in the process of making a protein, is taken to where the protein is being made at the ribosome and attaches that amino acid one after another to make the protein. And that process is called translation. And you can see on the far right-hand side all of the amino acids that exist, and they're each given a different letter of the alphabet so that we can use scientific shorthand to talk about which amino acid it is, instead of having to write them out. As you can see, some of their names are rather long. This is important to understand, because the genetic sequence, which you see here in red, and the protein sequence, which you see in blue, are 2 different sequences. They're not exactly the same. And this graphic here is a testing developed by Jean-Claude Perez with Luc Montagnier in Paris, the experts we talked about earlier with HIV, where they have been able to weight with a master code the sequences of these nucleotides and their energies and their importance. And when that's compared, that's on the y-axis, the ordinate. When that's compared with the genetic base sequences on the x-axis, or abscissa, you can see what you end up with is a red graphic line for the nucleotide bases. The proteins show up as the amino acids, and they show up as the blue line. And so you can compare the genetic code and the protein code using this specific approach. And what we show here in the circle is that prion-like domain that happens at the top of the spike protein. And you need to just carry that in your mind, because this is a very dangerous part of both the genetics of what's gone into this bioweapon and the actual structure, the prion-like domain of this bioweapon. And here's why it's important. In the upper left-hand corner, you're going to see the actual genetics in red and the protein structure in blue of the actual SARS CoV2 Wuhan spike protein. To the right of that, you're going to see the Moderna vaccine. And you will notice that the red line, the genetics of what's in the Moderna vaccine, does not match the genetics of the actual virus. There's a lot of similarity in the blue part of that, of the Moderna spike protein. As you get farther to the right, there's more variability. But the one area that is spot on happens to be that W prion-like domain. If you drop down, you're going to see the Pfizer vaccine. Again, the red shows the genetics that's in the mRNA of the Pfizer vaccine that they've talked about. And you'll see that that matches even less the spike protein of the actual virus. Remember, what we're being told is that what's in these vaccines are identical to the genetic mRNA of the spike protein. And in neither case is that true. But again, when you look at the spike protein that's produced, there's an awful lot of similarity with it being exactly the same at the prion-like domain. So these two vaccines do not genetically match the virus, even though we've been told that they do. The protein, the spike protein, comes pretty close, and it is spot on for the damaging prion-like domain. This also allows us to track all the mutational changes that are going on around the world with all the concern that people have for the variants. And you can see on the far left-hand corner there a variety of the different variants that are being talked about: V1, V2, V3, and Delta. Delta showing up in green, and you can see in a variety of countries. And if you go to this https covariance.org per country website, you'll find these and many more countries, as we are able to use this PCR testing to track these variations that are going on. And you'll see that, yes, the Delta variant is becoming more prevalent across the world, and particularly in the UK, the USA, and now in Sweden, Netherlands, and Italy and France. This is useful because understanding these variants are teaching us a couple of things. First off, as we look at them, we can either compare them based upon the commonality or how much is shared between viruses of these mutational changes. And we can also look at the commonness of this in the far right-hand side. And as you track down here, you can see these aren't unknowns. We know where these variations are occurring in the spike protein molecules of these coronaviruses as they are occurring throughout the world. And you can go to this site. This is the result of work not only by Luc Montagnier and Jean-Claude Perez, but also Emma Hodcroft, who's also a professor. And here's something that's very critical to understand about these mutational changes. If you go to that site, you will be able to look at each one of the shapes of these spike proteins, as we're seeing the protein model on the far right. But you'll also be able to, as we see on the left, see exactly what these changes are. And what's interesting is that while there are some increases in infectivity, the changes that are happening to these viruses show that these viruses are genetically changing the inserts that were made. They are working to remove the inserted components of the furin cleavage site and the HIV insertion sites. Now, an organism to survive when something is done to it that's damaging will attempt to repair itself and go back to baseline so it can survive. And this data provides evidence that the viruses, while everybody is concerned about these variants occurring, indicate that these viruses are attempting to repair themselves and go back to their pre-damaged state. As the mutations occur, as these spike proteins change, they're getting further and further from the SARS CoV2 that the vaccines were developed for. And as a result, they are becoming less and less likely to address the new variants because the spike proteins are changing enough that the vaccines that were given to people will no longer have any benefit. And we're seeing that already with the Delta variants. In fact, the mutations here on the Delta variant are occurring in the N-terminal domain, the receptor binding domain. Remember that prion-like domain, except the vaccines continue to carry the prion-like domain, and the spike proteins that are made from it with their prion-like domains that are not staying at the site of injection, but getting away from the site of infection where they're then able to cross the blood-brain barrier and cause prion-like diseases and get to the heart and cause weakened hearts or cardiomyopathies. And the furin cleavage sites, all of these are changing in these variants, in the mutations. These are the 4 critical ones that everybody's talking about. And it's important to know that there's 17 different variants right now, if you look on that left-hand column, and 12 different specific mutations occurring independently. And what you're seeing in those 4 examples in V1, V2, V3, and Delta, every one of those colored spots are areas where these viruses are attempting to change themselves, and they're the areas where the inserts were placed. Appreciating that PCR allows you to identify what you're looking at, and understanding that when genetic sequences match, it means you're looking at the same thing. This is extremely important. And it's extremely important specifically when we're asking about the origin of SARS CoV2 and its gain of function. These genetic sequences can be compared when you have them, and I do, using something very simply known as SnapGene. It's a viewer that allows you to compare genetic sequences over 30,000 kilobases, which is what this virus has. And when you do that, you can ask some very specific questions about very specific viruses, including these 3, which are Barrett's 3 chimeric viruses that we talked about at the beginning of this presentation. The RS3367, the SHC014, that was the spike protein to the backbone of another with the 5 inserts, and SARS-CoV-M15. When we ask a very fundamental question, what are these viruses? And we use PCR as the forward primer and reverse primer to code for them. What we see is that the SARS-CoV-2 virus matches Barrett's 3 viruses. His 3 gain-of-function research viruses have the same forward and reverse primer match sites as SARS-CoV-2. Coupled with this letter, published, including authors Peter Daszak of EcoHealth and Shi-Zheng Li of the Wuhan Institute of Virology, you can see that these viruses, the C015, the RS3367, which are funded by Foundation of China, the National Natural Science Foundation of China, the NIAID, Health and Human Services, USAID, and the federal government, you can see that this research, including these viruses, were paid for, in fact, with gain-of-function money. And it included not only Ralph Barrick and Shizeng Li, but Peter Daszak and all of our federal agencies in the United States. Including at the very bottom, you'll see if you want correspondence on this research, you're supposed to send it to Peter Daszak at EcoHealth or Shizeng Li at the Wuhan Institute of Virology. It becomes very hard to say that all of this is not connected when they provide you paper trails, and we now have the genetics that match all of this, these viruses, this funding, and these research papers. Let's turn now and take a quick look at how effective the vaccines really are and whether they're even needed. A lot of people use the term relative risk reduction or vaccine efficacy as they've come to know it, and that's a relative decrease in somebody developing COVID or being diagnosed with it. More critical is something called the absolute risk ratio, which is the actual difference between people that are vaccinated who aren't. And you can also look at the number of people that are needed to vaccinate, which is nothing more than 1 divided by that absolute risk ratio. And when we do that, we can look at the 3 current vaccines in the United States. We can look at the Pfizer vaccine and ask, using the emergency use authorization documents themselves, which provide us information about the number of people that were included in the study, the number of people who got vaccinated, the number of people who didn't get vaccinated, and the number of people who actually then were diagnosed with COVID. And using this, without going through it in great detail, but I'll leave it here for a moment so people can refer to it and then go get those emergency use authorization documents, which you can find on FlemingMethod.com. And when you do the math, you'll discover that the absolute risk reduction between the 2 groups showed that 0.93% of the people who had no vaccines were eventually diagnosed with COVID. And of the people that got the Pfizer vaccines, 0.05% of those eventually were diagnosed with COVID. And the absolute risk reduction for that is 0.88%. When we do the same thing with the Moderna vaccine, again, taking the Moderna emergency use authorization data in the documents filed with the FDA, and you look at how many people were in the vaccinated group and the non-vaccinated group, and you ask how many people actually ended up being diagnosed with COVID, what you discover is that in the non-vaccinated group, only 1.33% actually ended up being diagnosed with COVID versus 0.08% in the Moderna group, which is an absolute risk reduction of only 1.25%. Janssen or Johnson & Johnson wanted to report 2 sets of data. The 14-day data is the one that was given the emergency use authorization. Again, this is not vaccine approval. This is the emergency use authorization. And at 14 days, doing the same comparison, there was a statistical difference between the number of people who ended up with COVID who didn't get vaccinated, 2.38%, and the number of people who came down with COVID diagnosis who got vaccinated, 0.81%. That statistical difference was actually 1.57%, as you see at the very bottom. That was at 2 weeks after the Janssen or Johnson & Johnson vaccine. However, when 2 more weeks went by and they compared the data, that difference was no longer statistically different, and it was only a 1.01% difference between the 2 groups. between the non-vaccinated and the vaccinated group. And again, this is all this table I'm using here is in the emergency use authorization. Every one of those is in those EUA documents, which you can download from FlemingMethod.com towards the end of it, where we talk about the viruses themselves, along with 141 published papers, including about 30% of what I used to develop our research study, so that physicians and scientists and the general public can look and see there's actual published data showing drug treatments that work for these viruses, looking at the masks, looking at the vaccines, looking at everything you need to know. And it represents, again, about 30% of what I used to develop the research study. When we put all this together in a graphic table, you can compare the Pfizer, Moderna, the Russian Gamaleya, the Janssen, and the AstraZeneca, and look at either relative risk reductions or, more important, the absolute risk reduction, which goes anywhere between 0.84 and 1.3%, which is not, I think, what most people are thinking is really happening. It's really the elephant in the room that shows that the vaccines don't really have the efficacy or effectiveness that people believe they do compared with people who aren't vaccinated. Let's look at treating this bioweapon, because it's important to realize that not only are the vaccines not super effective, but the virus is quite treatable. There's 4 basic things to look at, and they include the attachment and the ability of the virus to replicate itself, which is shown inside this schematic. The ability of patients to get oxygen into their lungs and adequately survive. This is the acute respiratory distress syndrome component of it. And within the lungs themselves, or inflammation and blood clotting occurring, that's an inflammothrombotic response, which takes you back to my original theory that I presented in 1994. When the virus comes in, and you can take any of these viral sites, which are shown on the outer surface of this cell, it requires 4 receptor sites: the ACE2 receptor, another receptor called transmembrane protein series 2, TMPRSS2, the furin cleavage site, which is back to that PRRA, and the neuropilin site, particularly important for the brain. Once it gets into the cell and it's reproduced, or whether it gets in through a vaccine, as you can see on the either the lipid nanoparticles on the bottom left, or the adenovirus to the left of the cell, and it gets inside, it begins the process of making the proteins. Those proteins are initially, whether they're a complete virus by person-to-person transmission, or the spike protein by the vaccines. In many instances, those get broken down by proteins inside the cells called proteasomes, and they will get first presented to the cell surface in a matter of days from major histocompatibility complex 1, and that exposes the bits and pieces of these products to our T cells, which begin the first part of our immune response, and that produces inflammation and blood clotting, which has to be controlled. When either the spike protein or viruses get outside the cell, they'll be taken up by other cells and undergo that same process, except then they get presented to cell surfaces by what's called major histocompatibility complex 2, which allows B cells to start making antibodies. And it's not so much the chemicals produced by the T cells or the antibodies produced by the T cells that you want over the course of time, but you want the memory cells, because your body's not constantly releasing these damaging chemicals or blood clotting or antibodies, because that would be a waste of energy and would actually make your blood very thick and make it impossible for you to survive. What you really need to know is whether you're making memory cells, not so much whether you still have antibodies floating around. All of that being said, every one of these parts has to be addressed, and this is what the treatments need to focus on. At the very bottom, you'll see one of the papers that we've published showing successful treatments of people who ended up with COVID. successfully treating them 99.83% of the time. So these are the worst outcome people. Jokar just published a paper in Virology Journal talking about the use of PCR testing and various samples from the body, including the nose, the blood, the urine, and stool. And they specifically noted that this is very critical and shows treatments that work. And the 3 papers that they quoted for treatments that work were the 3 papers that we have published to date. So they're recognizing that the treatments work as well as the research that we've done and other individuals working with us. Here are the treatment regimens. For people that haven't been infected with the virus yet, you can do things to improve your immune support in addition to doing all the other things like getting your weight under control and not overeating and getting the weight down and working on your heart disease and diabetes, but you can also provide immune support to enhance your immune system. That entire regimen right there, which is available for people now on FlemingMethod.com or for people watching this video, is a whopping $33 in the United States per month. Certainly not an overly extravagant regimen. Once people start to become symptomatic, this approach increases the amount of those vitamins and minerals that they're receiving, and it's just under $50 a month. And we use these regimens in people in the research study that were symptomatic, both in the outpatient and hospital settings. In the outpatient setting, we have actually broken down what the different drugs are. And you can see treatment 1 was 100% effective. Now, if we get enough people with SARS CoV2, the virus infection, obviously treatment 1 can't work for 100% of everybody. It has so far, and that includes hydroxychloroquine, primaquine, and clindamycin. The doses are provided there for people, along with inhalers that will open the airways if needed, and that's for people with oxygen saturations of less than 94%. And the total cost of that regimen is $95. The treatment that was second most effective did not include primaquine, but had the hydroxychloroquine, clindamycin, ipratropium, or atrovent, and that was $46. And you can see treatment 3 was 74.2% effective. This is the one that everybody thinks of with the hydroxychloroquine, azithromycin. That cost a whopping $40. And the last one was hydroxychloroquine doxycycline, again with the inhaler when needed, and that was $40. So that was the outpatient setting treatments for people. Inpatients, this looks a little bit more complicated. It is on the website, but it has 2 things here. To the left-hand side shows what happens for the people who ended up being diagnosed with the disease COVID. COVID is not the virus. COVID is the inflammation and blood clotting, the inflammothrombotic response that occurs in response to the virus in people with the comorbidities of advanced age, heart disease, obesity, diabetes. And these sequences work 99.83% of the time. So there's either a combination of methylprednisolone or combinations with tocilizumab and interferon alpha 2 beta, or if they hadn't had something like primaquine or hydroxychloroquine as an outpatient, they needed to receive it inpatient, which is treatment number 3. And you can see the cost down below. And in each of these cases, you also note that we gave heparin and aspirin and the inhaler for the people, or nebulizer when needed. And this is important because we wanted to decrease the blood clotting associated with an inflammothrombotic response. And you're going to see in a moment how extremely important that is. The part on the right shows putting the outpatient and inpatient flow chart together for clinicians, physicians wanting to know exactly what to use and in what sequence. Finally, for the people that have been vaccinated that are having consequences, or the people that have been exposed to people who've been vaccinated with consequences, shedding, which can't be ignored anymore. In the event 2021, I shared with people the data that FDA and Health and Human Services provided in 2015 or possibly 2017 as information for the industry, where they talked about shedding from RNA and DNA gene therapy vaccines, their term, not mine. They actually shared that with the industry. This shows the multiple step process for addressing vaccine and shedding consequences, beginning with protecting the nuclear material in our cells in step one, to attacking the actual spike proteins floating around our bloodstream, which we know occur, to interfering with the uptake of those spike proteins across the blood-brain barrier, to then further treating the actual prion-like disease itself. And you'll notice here that at that part, we're talking about medicines that have been used in cardiology for some time for lipid disorders, along with heparin, or if you don't have it, enoxaparin and aspirin. That's important because heparin interferes with the ability of this virus to attach itself to our cells. It interferes with the spike protein. So, and it does so better than enoxaparin. So good old-fashioned heparin turns out to be the Achilles tendon of this human-engineered gain-of-function bioweapon. It actually interferes with the ability of the spike protein to attach, and this was published in 2020, as you can see, by Mycroft West and others. I think it's also important to address another elephant that's in the room, which has to do with natural immunity versus these vaccines. All three of the emergency use authorizations approved in the US admitted that they had no information about any benefit for vaccinated individuals who had already been exposed to the virus. On the left-hand side, you can see a study that was published by Nielsen recently this year, where they're looking at both IgG, and if you've listened to me long enough, the importance of IgA antibody, which is associated with viruses that infect the respiratory tract and GI tract, like SARS CoV2 does. And what you can see there is that people who were exposed to the virus by person-to-person transfer had antibodies that were made to the full-length spike protein, to the regional binding domain of the spike protein, to the N-terminal domain of the spike protein, to the nucleocapsid of the virus itself, both IgG and IgA, and those antibodies went up as the severity of the infection was worse. But it's important to note that it didn't matter how severe the infection was, these antibodies were protective and measurable. And on the right-hand side, you can see that the T cells, in response to other influenza viruses and cytomegalovirus, something that many people have had, that the study on the right that was done by Mahayan also points out that we have T cell immunity to SARS CoV2 if we've been exposed to influenza or cytomegalovirus. So we don't need to just have been exposed to SARS CoV2. We could have been exposed to these other viruses, and we've made T cell responses to these other viruses, which is protective against SARS CoV2. Finally, I want to point out the discussion that people seem to be confused about as to whether this virus actually gets into human DNA. The glycoprotein 120, I originally thought that Shi Zhengli inserted into it because of its reverse transcriptase, or its ability to get back into human DNA. Shi Zhengli was doing that work at the same time that the Human Genome Project was being completed. So we didn't know at that point in time that 18% of the human genetic code has what's called long interspersed nucleotide elements 1, which are our ability to take genetic material from outside of us and put it into our DNA, or reverse transcribe. Remember, transcription is DNA to RNA, so reverse transcription is RNA to DNA. We know that this genetic material from SARS-CoV-2, and it's independent about whether it's coming from the person-to-person transmission of this virus or the vaccines, that it has been shown now as of this year to reverse transcribe or insert itself into all but 3 human chromosomes. And you can see them listed here. Of the 23, only chromosome 8, chromosome 15, and chromosome 20 have not been associated. And you can see on the far right, human chromosome X, that's the male chromosome, insertion of the genetic material from this mRNA virus, SARS-CoV2, into human DNA. The ultimate bioweapon isn't a virus or anthrax or fungus or a bacteria. The ultimate bioweapon is a prion. Hard to treat, until recently impossible to treat, except for this interesting information about heparin and the research results that we saw, and now some of the cholesterol lowering medicines that have shown promise in these prion diseases. It's not only been previously untreatable, it's been slow and malicious and progressive over time. So that exposing your enemy to this not only devastates them when you see what happened in 2019, 2020, and even 2021, as far as shutting down societies and alienating people and causing mass confusion and damaging economies and putting people out of work, but it's a slowly progressing prion disease that causes neurologic disease, brain damage, heart disease, which we're seeing in younger individuals, slowly, progressively over time. The bottom left shows how these prion proteins distribute themselves between nerve cells. These red areas show the infectious prions moving from one neuron to another. To the right of that shows the spongiform open brain spongiform encephalopathy or mad cow disease that's been associated with brain shrinkage. And it's a slow progressive process that occurs by constantly re-ingesting these prions. And you can either get them from the person-to-person transmission, or they're the very proteins that are being made by cells from the vaccines. Our last area, what I call Nuremberg 2. These bioweapons were released. They were paid for, engineered, designed, built, and released, causing tremendous harm intentionally. I would say the world stage equals a large scale event. And by definition, they are crimes against humanity, and anybody associated with the development, funding, design, building, dissemination are guilty of crimes against humanity. They have built a biological weapon that is in violation of the Biological Weapons Convention Treaty, which bans any of these weapons by anyone at any time. I'm including slides for your viewers and for people to see of these actual biological, of every one of these violations, including the 1975 Biological Weapons Convention Treaty. I want this to be available for everybody, for everyone to use, to share, to take to attorneys, to take to courts, to take to those responsible, to show these violations. You can read through them at your leisure. There are, for the Biological Weapons Convention Treaty, 15 articles. And those are the nation states that have signed and ratified, including Russia, and China has acceded or seceded to it. The United States, all the parties that are involved. Above and beyond the United States and China, I might add. We're just beginning to warm up on the parties responsible. The 1947 Nuremberg charges are exactly the charges that I'm calling for for 2021 Nuremberg 2.0 charges. In the first round, there were 24 defendants of the first part of the Nuremberg trials. The 4 crimes were crimes of conspiracy. They worked together in what they did. Crimes against the peace, intentional acts of war, war crimes. They've intentionally harmed civilians, and crimes against humanity. Harm against all people around the world, resulting in death, inhumane acts, damage, harm, a variety of harms. In 1947, Goering blatantly told the tribunal that any country could get any people to do whatever they wanted if they simply told the people that they were under attack, and a failure to do what they were told to do was anti-patriotic. In 1947, the first set of trials from Nuremberg had 24 defendants. It would have had 26, but Adolf Hitler and Joseph Goebbels committed suicide before they could be held accountable. And you will see the names of these different individuals and the positions that they held in government, as well as their sentencing and actions taken. In none of these individuals' cases were they given their requested method of death if they were condemned to death. Many of them, like Wilhelm Keitel, wanted to be shot as a soldier. They denied that and hung him. He didn't deserve the death of a soldier. What he did was not the actions of a soldier. Of these 24 individuals, 12 were given a death sentence, 3 life imprisonments, and 4 were sentenced to 10 to 20 years. There were 12 trials for Nuremberg. This was the first one. 11 others followed, and it began with the doctor's trial. As a doctor, I'm calling for the doctors involved with this to also face charges. During the Nuremberg trial, there were 23 doctors charged. 7 of them argued that the defense of what they did was that they had done nothing worse than other American doctors or German doctors had done, showing that there weren't any international or informal statements about illegal treatment of people, resulting in doctors Andrew Ivy and Leo Alexander feeling compelled to make a statement so that these doctors did not get off from the harm that they had done to people. And as a result, they came up with 6 points that they argued as permissible for medical experiments. Those 6 points got expanded to 10 and became the 1947 Nuremberg Code. Here's a page from volume 2 of that, old and aged. The 1947 Nuremberg codes are very specific about what permissible medical experiments could be done. And in every instance, it was absolutely essential that the people that participated had informed consent. And that prior to experimentation upon humans, that animal research had to be conducted. Every effort was being taken to prevent harm to human beings. The protagonists in these trials justified what they were doing, stating it was necessary for the German people, to protect the German people, necessary for their society. The 1947 Nuremberg Code laid down 10 standards that physicians must conform to. This is accepted worldwide, and voluntary informed consent is a requirement for the human subject, for any human being to know what's going on. These next few slides show in detail what these 10 codes are. And they're here for you to look through and to share and to do the same thing, to hold people accountable. We are being held in history to determine if we are going to respond in the same way that our ancestors did with Nuremberg 1, or whether we're going to allow these crimes to go unpunished and deal with the consequences for our children and grandchildren. This provides you an opportunity to find these. Sources are listed on the very bottom of this, so you can go look them up on your own, and to share and begin a movement to hold these people accountable. The doctors' trial that followed these 10 codes considered the fate of 23 doctors. Thanks to the work of the 2 doctors demanding a Nuremberg Code, 16 of the doctors were found guilty and 7 were executed for crimes against humanity, for violating their oaths. To give somebody a test or a treatment or an experimental drug without providing informed consent is a violation of the American Medical Association Code of Ethics. And I'm certain that it is for the medical associations throughout the world. But this one in particular, in the United States, that it's important that informed consent be provided. It's both medical and legal, that it's between the patient and physician, and it requires an explanation of the risks, benefits, and the purpose of these experimental procedures. And it's important that we look at the package inserts, because as a physician, we know what the risks are, either because we look in the physician desk reference, which is nothing more than a conclusion of all the package inserts, or the package inserts. This is the Declaration of Helsinki. And within the Declaration of Helsinki, there are multiple principles that physicians are held to in the experimental treatment of patients. These slides, again, can be looked at over the course of time for you to be familiar with. But here's the important point about both the Declaration of Helsinki and the American Medical Association Code of Ethics. To the left, you will see the package inserts of the Pfizer, Janssen, and Moderna vaccines. They are empty. We opened these at Event 2021 to show to people, and I reproduce them for you here. If you are a physician who's administered or ordered a SARS CoV2 drug vaccine, and you've not read the emergency use authorization documents, then you have violated the AMA Code of Ethics. If you have read the emergency use authorization documents and ordered the SARS CoV2 vaccine, then you have violated your physician's oath. You have not provided informed consent to people, and the doctors, like the politicians and the government agencies responsible for this vaccine and this bioweapon, should be held equally accountable in criminal court. This is the Hippocratic Oath, and this is the Physician's Oath, a Declaration of Geneva, my oath that I took. I'm going to read it for emphasis. At the time of being admitted as a member of the medical profession, I solemnly pledge myself to consecrate my life to the service of humanity. I will give to my teachers the respect and gratitude which is their due. I will practice my profession with conscience and dignity. The health of my patient will be my first consideration. I will respect the secrets which are confided in me. I will maintain by all the means in my power the honor and the noble traditions of the medical profession. I will not permit considerations of religion, nationality, race, party politics, or social standing to intervene between my duty and my patient. I will maintain the utmost respect for human life, even under threat. I will not use my medical knowledge contrary to the laws of humanity. I make these promises solemnly, freely, and upon my honor. Violation of this with this bioweapon is a crime against humanity and a crime against our oath. It is also a violation of the International Covenant on Civil and Political Rights under Article 7 that no person shall be subjected to cruel or inhumane punishment, and in particular that no one will be subjected without his free consent to medical or scientific experimentation. And this particular treaty has 53 articles in it, of which Article 7 here is the one that I just read to you, but every one of them pertain to our civil and political rights established after World War II to make certain that these would never be violated again. This is Dr. Joseph Mengele. He came from a well-to-do family. He was not in charge of Auschwitz, but he was an SS doctor who received lots of German government grant money to study genetics and diseases. Sound familiar? Lots of federal money to study disease. In the bottom, you'll see him riding a bicycle in Argentina. A good friend of mine's family was hunted down by him and someone that I actually trained with at fellowship. The people on the bottom, these were the citizens of Germany. Except instead of just experimenting on people in prisoner of war camps, SARS CoV2 was an experimentation that continues today around the world on all people of all faiths, all religions, all creeds, still being conducted. I'm calling for a second Nuremberg. And these people here in this list are only the beginning. They should all be investigated, indicted, prosecuted, and held accountable, not only within your own countries, but before the International Criminal Court, for crimes of conspiracy, crimes against peace, war crimes, and crimes against humanity. For the development of a biological weapon in violation of the Biological Weapons Convention Treaty. For the testing of a biological weapon in violation of the Biological Weapons Convention Treaty. For the release of a biological weapon in violation of the Biological Weapons Convention Treaty. For the interference with the practice of medicine, prohibiting physicians from prescribing medical treatment to those who were infected with the development and released biological weapon. For the interference with the practice of medicine, prohibiting physicians from prescribing medical treatment to those who develop the inflammatory thrombotic release disease and subsequent disease COVID-19, resulting from the infection with the developed and released biological weapon. For the failure to follow the required testing of drugs in tissue and animal models prior to giving an experimental drug vaccines to humans as required by the Nuremberg Code, Declaration of Helsinki, the International Covenant on Civil and Political Rights, and the AMA Code of Ethics, and applicable Code of Ethics in other countries. For the failure to obtain informed consent before testing experimental drugs under these same codes. For experimentation upon citizens, including the frail, elderly, and those who are physically and mentally disabled, using these experimental drugs in violation of these same codes. For experimentation upon our children, using experimental drug vaccines in violation of these same codes. For courts, for judges, who have carried through experimentation upon prisoners and defendants, as seen here by this judge who required as part of the punishment that those found guilty had to undergo experimentation with these drug vaccines. For the loss of personal liberties, income, assault, threat, and coercion of citizens to participate in these experimental drug vaccines, following the suppression of treatments and failure to follow required testing of drugs, including drug vaccines in tissue and animal models, prior to giving these experimental drug vaccines in violation of the Nuremberg Code, the Declaration of Helsinki, the International Covenant on Civil and Political Rights, and the Code of Medical Ethics by the AMA and other applicable medical associations in the world. For the loss of personal liberties, income, assault, threat, and coercion of citizens around the world, including forced quarantine, lockdowns, and masking, and for the subsequent morbidity or mortality associated with the development, release, and testing of this biological weapon in violation of these treaties, codes, and ethics. These individuals should be brought to International Criminal Court, and you can see here the nations that have signed and ratified. In the United States, I would encourage us to have Congress ratify what has already been signed. The Europeans have done so. And if we need to take this to International Criminal Court in The Hague, I believe it is well past time for us to hold these individuals, both those accountable for funding and developing and releasing, but for those responsible for further disseminating further biological weapons, because recognizing that the gain of function bioweapon is a prion disease carried by a virus that is man-made, vaccines that code for that same spike protein bioweapon are also bioweapons. If you've ever wondered whether you would have been compelled during the 1990s, 1930s Germany, you now know. I think it's time for us to prove Hermann William Goering was wrong when he said that societies, governments could manipulate their people. I think it's time for the people to stand up in every country around the world and demand justice and a cessation to this experiment upon humanity and these crimes against humanity. There's more information available just on the virus, as well as to let you know that on September 7, we are releasing this book with much more information about those involved in this bioweapon, the funding, the papers that were published, the patents that were given, and the relationships. Is COVID-19 a bioweapon? A scientific and forensic investigation. And with that, I'd like to thank for being invited for this conference and this presentation.
Dr. Fleming, this is an absolutely outstanding presentation. I'm humbled and I am grateful for you providing this information at this conference. And we shall transmit this far and wide, and I will get on with what is required to make this happen.
The video that you gave us that I posted on the website has had a huge impact on people. You need to know that you're making a huge impact on people. It's resonating with them, and they are finding this extremely useful information. So if you ever wonder whether what you're doing is reaching people, rest assured it is.